In this lesson on the inheritance of qualitative traits, we focus on segregation and independent assortment, which are Mendel's first and second laws. And what I want to do, instead of just trying to have you memorize these things by brute force, I'd like to try to connect them with what you already understand about the mechanics of meiosis. It all fits together as a system, and I think it's easier to remember if we can see how these connect rather than just try to memorize them as completely separate mechanisms. So let's start out by taking a look at the inheritance of qualitative traits, where we're just looking at one trait at a time. In the lesson, I talk about Mendel's P's, and in particular the inheritance of smooth versus wrinkled P's. So instead of going through the same treatment that I use in the lesson, let's take a look at this as if we are working through meiosis. So in the inheritance of smooth versus wrinkled, what Mendel did was cross a plant that was smooth with a plant that was wrinkled. So let's start with the parents. One of the parents, the smooth parent, is going to look like this. It's going to have, remember, two homologous chromosomes that are going to carry the gene for smooth or wrinkled. And in the case of the smooth parent, both of the homologs are going to have the capital S, the dominant gene for the smooth P. So if this is, let's say, the male parent, just arbitrarily, we're going to have one of the homologs is going to carry the big S allele at the locus for the seed smoothness, and the other homolog is going to have that same capital S allele. It's going to go through replication and form sister chromatids. At which point our chromosomes are going to look like this. So now we have the sister chromatids and we're heading into meiosis. Meanwhile, on the female side, the same thing is happening. But let's say where the male is big S, the female is our wrinkled parent, and it's going to have genotype little s, little s. So its homologs are going to look different because it's going to have the little s allele at the locus for seed smoothness or wrinkledness. And when it goes through replication prior to meiosis, now the sister chromatids look like this. Well, getting back to the male that's homozygous dominant, when all is said and done and meiosis is over, all of the gametes are going to be identical that come out of this parent. They're all going to look like this. This is our sperm. We're going to have this particular chromosome, and the allele on there is going to be capital S. All the gametes from the male side are going to be capital S. It's the only possible. Look at all these sister chromatids. All they are capital S. So that's the only thing that could be in the gamete. In contrast, when we take a look at the homozygous recessive, the only possible allele in the gamete from this parent is going to be the small S allele. Because when we look at the sister chromatids, that's all that's there. So when we cross the homozygous dominant big S by the homozygous recessive little s, then the F1 is always going to be the combination of these two particular gametes. And so the zygote is going to have the big S allele from, in this case, the male, and the little s allele from the female, and so it's going to be the big S, little s heterozygote. Now things get a little bit more interesting when Mendel self-pollinated this F1. So let's see what happens there. So in this case, looking at the male again, it's big S, little s, and the female is also going to be heterozygous, big S, because again, what we're doing is selfing the F1 that we just created that was a heterozygote. So when you're selfing a heterozygote, it means the male's a heterozygote and it's the same plant, the female is also a heterozygote. So now the question is, what sort of gametes can be produced? Well, again, if we take a look at what we have in this cell, we have a chromosome that has a big S and a chromosome that has the little s. And they're going to go through replication. So that now we have the two homologs where these two sister chromatids are both big S and these two sister chromatids are little s. Now exactly the same thing happens over here when the female is undergoing meiosis. It's going to look identical because the genotype is the same. Big S for one of the chromosomes, and because it's a heterozygote, little s for the other two sister chromatids making up the other homolog. Now, what sort of gametes do we get from this? Well, there are two types of gametes that can result from meiosis here. One of the gametes is going to be a big S allele gamete, and the other one is going to be a little s type of gamete. And they're in equal proportion, equal frequencies. So these are the two types of gametes that result from meiosis in this cell. And because these two are identical, then this cell on the female side is going to have the same type of gametes. So it's going to have a gamete that has the big S allele and also gametes that have the little s allele. So these are the two types of gametes on the male side and the same two types of gametes on the female side. Now the question is, what sort of progeny, what sort of offspring are going to result? And this is where we use the Punnett square. And in the lesson what I did was put the types of gametes from the male side on the top of this Punnett square. So one of our gamete types is S, and the other one is a big S and small s. And I put the female gametes that I'm just erasing down the left-hand column. One of the gametes was big S, and the other was little s. And I arranged this as a bit of a database. And then we can do the matings. We have a male gamete that's big S and a female gamete that's big S. The zygote that's going to form is the homozygous dominant. If the male is small s and the female big S, then it's going to be heterozygous. Likewise, if the female is small s and the male's big S, it's also going to be heterozygous. You notice that when I put this notation down, it doesn't mean that one is from the male and the other is from the female. You always put the capital first, just by convention. And then if you have a recessive S female gamete with the recessive S male gamete, you get the homozygous recessive. So that's how you use a Punnett square. You put the gametes from the male on the top, the gametes from the female along the left-hand margin of the rows. They're in equal frequencies, and then you can figure out the genotype ratios and the phenotype ratios. So the genotype ratios are for big S is 1, the heterozygote is 2, here and here, and the homozygous recessive is 1. So the genotype ratio is 1 to 2 to 1. This is genotype. But when we ask what's the phenotype ratio, and that means what do the seeds look like to us, then remember these three categories all look the same because the capital S is dominant over the small s. So these look the same as this. So the phenotype ratio, phenotype, is smooth equals 3 and wrinkled equals 1, or a 3-to-1 phenotypic ratio. So the idea here was to take a look at Mendel's first law of segregation, in which we find that when we have two alleles, one of those alleles is going to be found in one gamete and the other allele in another gamete. You never find those two alleles together in the same gamete. They split up into separate gametes. They segregate. And when we follow that through meiosis and take a look at what happens in the gametes and how they then come together to form zygotes, then in this case we have a genotypic ratio of 1 to 2 to 1. The 1s are the homozygous classes, homozygous dominant. The 2 is the heterozygote, and the 1 on this side is the homozygous recessive. But how they look, the phenotypic ratio is 3 to 1, with 3 smooth and 1 wrinkled.